A nucleotide of a DNA molecule has three components. A 5-carbon sugar that is either a deoxyribose sugar or a ribose sugar, a phosphate group, and a nitrogenous base. There are 5 bases: A, T, C, G, and U. U, or uracil, is found only in RNA, and T, or thymine, is found only in DNA. Adenine and thymine pair together, and cytosine pairs with guanine. The two strands of a DNA molecule run antiparallel, as one runs in the 5' to 3' direction and the other runs in the 3' to 5' direction.
Thanks to the Messelson and Stahl experiment, it was known that DNA replicated in a semi-conservative manner. The semi-conservative model represents that when one DNA molecule is replicated, the two new daughter DNA molecules have one newly synthesized strand of DNA and one old parental strand.
Helicase unwinds and separates parental DNA strands, forming a replication bubble. This continues unwinding at the replication fork, creating a Y shape in the DNA.
Single-strand binding proteins, also referred to as SSBPs, stabilize the unwound parental strands and keep them from reattaching to each other.
When DNA strands are separated by the helicase, it forms a leading strand, which is elongated continuously in the 5 to 3 direction as the replication fork progresses. There's also a lagging strand, which elongates in the 3 to 5 direction. This is synthesized discontinuously.
RNA primer is a short RNA chain consisting of ribose sugars as part of the nucleotides, making it RNA nucleotides. These initial nucleotides are added only to the 3' end as DNA is synthesized, and only one nucleotide is added at a time. Primase is the enzyme that synthesizes these RNA primers, and it uses the parental strand from the semi-conservative model as a template for these primers.
After RNA primer runs its course, a total of 2 bases are added. DNA polymerase III adds one nucleotide at a time to the leading strand. This and then creates phosphodiester bonds between each nucleotide.
Because the lagging strand of the DNA is synthesized discontinually, the fragments on the lagging strand are known as Okazaki fragments, named after the scientists who discovered them. The enzyme DNA polymerase I replaces all RNA nucleotides. It does this by removing the primer from the 5' end of the lagging strand and replaces it with DNA nucleotides that it adds one by one to the 3' end of the strand.
DNA ligase joins the 3' end of one fragment to the 5' end of another fragment. It joins the sugar phosphate backbone of all Okazaki fragments into a continuous DNA strand.